Hello, I am Mark Hopkins, and I am with the Wild Peanut Lab on the campus of the University of Georgia in Athens, Georgia. Our poster describes the genesis and characterization of interspecific hybrids to increase the genetic variation in cultivated peanut. The rationale for this undertaking is that peanut underwent an event 8 to 10,000 years ago that created a genetic bottleneck for the species. An interspecific cross between two wild species, followed by chromosome doubling, produced the tetraploid cultivated peanut we know today. There is a large amount of genetic variation in wild peanut species. Because nearly all wild species are diploid, this event severely limited the amount of genetic variation in cultivated peanut. The Wild Peanut Lab utilizes a proven pipeline to introduce the genetic variation found in wild species into cultivated peanut. As seen in the pipeline illustration, we cross wild peanuts to create a sterile diploid hybrid, treat with colchicine to induce tetraploidization, then cross with cultivated peanuts to incorporate the new genetic material into the genome. The next steps are a series of back crosses with the recurrent parent to remove any unwanted phenotypic traits. This is followed by selection via the use of genetic markers and field trials, which eventually lead to improved peanut varieties. This presentation is limited to the initial crosses within this pipeline. Step 1 of this process is the selection of wild species to produce the interspecific crosses. A wild species containing the AA genome is crossed with a species containing the BB genome. The result is a sterile AB hybrid. These hybrids show a large amount of phenotypic variability. At the macro level, there are plant architectural differences from plants that are prostrate and plants with an erect main stem. There is floral variation reflected in color differences as well as flower shapes. There are also differences in foliar size and shape between the hybrids. Variation can also be seen at the cellular level with respect to pollen fertility and counts of stomates, epidermal cells, and trichomes. The chart indicates the amount of cellular variation that exists between the diploid hybrids. The bars represent each of the newly created diploid hybrids, with yellow indicating pollen fertility, green stomate counts, blue epidermal counts, and black trichome counts. As we expected, there is variation between diploid hybrids of differing species combination. But if you look at the first 4 hybrids from the top of the chart, created from the same plants but were differing crossing events, we can see that there is a large amount of variation between them. For instance, they range from 42% pollen fertility to 5% pollen fertility. They also vary greatly with respect to epidermal cell counts and trichome counts. It is important to make clear that this phenotypic variation seen reflects the potential genetic variation that can be incorporated into cultivated peanut. Step 2 of the pipeline process is to treat cuttings from these sterile diploid hybrids with colchicine to produce chromosome doubling and a fertile neotetraploid. Pictured are pegs developing from 2 of these hybrid combinations, and our newest neotetraploid from a cross between Erica's valida and Erica's microsperma. In conclusion, we have generated 26 new hybrid combinations, and 8 out of 22 colchicine-treated hybrids are producing pegs. There is not only a large amount of phenotypic variation seen between different hybrid combinations, but also within similar species combinations, which reflects the amount of genetic variation that can be introduced into cultivated peanut. Thank you for your interest.